Hi everybody, thanks for coming tonight. Our lecture today is Today's Junk, Tomorrow's Triumph, Lessons on Evolutionary Contingency, Inertia, and Innovation from the Long-Term Evolution Experiment with E. coli. I think if there was an award for the longest lecture title we've ever had, this would be it. So today's lecture is sponsored by the Michigan Humanists, and our speaker today, Dr. Zachary Blum. A little bit about Michigan Humanists first. We have a two-part mission. First is to advance a secular community. Secular means anyone identifying as an atheist, agnostic, humanist, skeptic, or a free thinker. And by community, we mean we share experiences, offer support, and engage in community service. We want to end the stigma attached to being a non-believer. Our second mission is to promote humanist values, those being reason, science, and church-state separation. More specifically, we oppose the influence of religion and pseudoscience in public policy, and we defend the rights of non-religious people. We have lots and lots of events. Monthly, we do Cafe Inquiry, Skeptics in the Pub, Living Without Religion, and the Sci-Fi Book Club. Quarterly or so, we do lectures like this, so there's about 3 a year. The summer, we take some time off to do one of our annual events, which is the Long Lake Retreat. Other annual events include tabling at the Pride Festival, which is coming up this May, and the Interfaith Thanksgiving celebration in November, and our most recent one, the Solstice Dinner this past December and every December. We also do some volunteering opportunities. The next one is going to be a Long Lake Work Day. That is typically in the late spring, April or May. So to stay connected with us, we hope you want to do that. You can visit our table right outside the lecture hall here, or sign up for our newsletter. You can make a donation out there or online. You can also see us online where all of our events and things are located. After the lecture tonight, we're going to have Q&A for 15 or 20 minutes or so. And if you would like, you can visit the Fountain Street exhibits. Looks like there's a quilting exhibit here. And then, I'm not sure what was around the church last time. It was a celebration of local women artists. So don't quote me on that, so you have to check that out too. And then we are going to go out to the Palace of India. That's 138 Fulton Southeast. And if you would like later, or you can tell a friend, you can listen and watch the lecture online. So let me tell you about our event tonight. Our speaker, Dr. Zachary Blum. His occupation is he is an assistant professor at Michigan State University and the director of graduate education innovation in the Department of Microbiology, Genetics, and Immunology at Michigan State. He has a BS in applied biology from the Georgia Institute of Technology, an MS in biological sciences from the University of Cincinnati, and a PhD in microbiology and molecular genetics from Michigan State. As a researcher on the LTEE, or as you'll hear, the Long-Term Evolutionary Experiment, his interests include how complex traits evolve, how novel traits impact ecology, and how speciation occurs in asexual populations. The LTEE is the longest-running microbial evolutionary experiment in history. It began in 1988 by Dr. Richard Lenski, also at Michigan State. Dr. Blum has been a part of that experiment now for over 20 years. His doctoral studies were with Dr. Lenski, and his dissertation was on the LTEE. He's also published on the LTEE, 16 peer-reviewed articles, written numerous articles for science magazines, and spoken to numerous groups. So with all that, please help me welcome Dr. Zachary Blum.
I will warn you and also reassure you, I have a lot of text on my slides. That's how I come up with my script. If you can't see the text, that's okay. I say pretty much everything that's on there. It's just how I work. So this is a really great thing for me. I really appreciate the invitation for many reasons. I love talking about this, but also we're gathered here to honor Charles Darwin. And Darwin holds a big place in my heart. He's genuinely one of my greatest heroes, because he's not only this person who was a fantastic scientist, but he was also a fundamentally decent human being, loved his family, cared about people, and managed to have tremendous productivity and change the world while being disabled with chronic illness that kept him in a very bad condition for much of his life. So he's truly an inspiration. And I was fortunate enough, in one of the highlights of my life, to get invited to the UK in 2023 by my good friend Brian Kloss, who's a journalist and writer. And we went around and saw a Darwin location. So got to see his statue at the British Museum of Natural History, got to walk his thinking trail, got to see his house. I got to stand in his study, which is set up just like he had it, and hold in my hand my copy of The Origin, the first one I read, because I read it many times in many different editions, and whisper to it, "This is where you were born." So I'm that level of nerd. This is just wonderful to me. So it is really right that we revere Darwin in biology, because he, through a lot of insight and a lot of thoughts that people just failed to have, and then 20 years of painstaking work, assembling mountains of evidence and showing how many diverse lines of evidence came together to show that life only makes sense if it had evolved. And then that is what went into The Origin. And in doing this, he turned biology from fundamentally an activity of stamp collecting of disconnected facts and turned it into a science. And he articulated the very first naturalistic explanation of how evolution could occur, and also explained how it is that organisms come to be adapted, so exquisitely fitted to their places in nature. So this is evolution by natural selection. Deceptively simple, and yet so easily misunderstood. You know, a population is made up of individuals that vary in their heritable traits. And we know now, Darwin did not know this, that traits are heritable because they're encoded in DNA sequences and through base pairs, and information gets transferred from generation to generation through these sequences, and then variation in traits arise because of mutations that change these sequences. And these mutations are random, so variation itself is random. And then the environment exerts selection on the population because the environment exerts stress. It presents challenges. And then also there are a finite number and amount of resources that are necessary for organisms in any population to be alive and reproduce. And so individuals compete for those resources. And then any individual with a heritable trait that improves the ability to compete for resources, this is to say it's adaptive. It allows those individuals possessing it to, on average, leave behind more offspring. So over time, that adaptive trait becomes more frequent in the population. And the population, not the individual, but the population evolves, with that trait becoming more frequent, and this happens over and over and over again. Beneficial traits pile up. Adaptation becomes better. Fitness increases, and over time there are massive changes in observable traits, so the phenotype of the organisms. As Darwin put it, natural selection is daily and hourly scrutinizing throughout the world the slightest variations, rejecting those that are bad, preserving and adding up all that are good. So we're 166 years on from The Origin. The work goes on. There are tens of thousands of scientists worldwide continuing to investigate all of these mysteries that remain in evolution, to unravel its complexities, some of which Darwin himself pointed out and we still haven't solved. And they use a variety of systems to do this. So they study fossils, they compare living things through the comparative method that he invented, they use digital evolution platforms to study digital evolution, and they take advantage, like me, of one of Darwin's great mistakes. Because Darwin also wrote in The Origin, we see nothing of these slow changes in progress until the hand of time has marked the long lapse of ages. So evolution is something that's always happening, but it's so slow we can't see it happen. As it happens, Darwin was very wrong about this. Evolution can occur very quickly. And ironically, there was evidence of this in his own lifetime. One of the people who read The Origin was William Henry Dallinger, who was an English Methodist minister, amateur natural historian, and also professional microscopist and biologist. He was president of the Royal Microscopy Society. And he got just fascinated by Darwin's ideas. He started exchanging letters with Darwin, and he said that he was going to undertake an experiment to test Darwin's ideas. And so he did. An experiment that lasted 7 years, from 1880 to 1886, so it overlapped when Darwin died in 1882, so he didn't get to see the end of this. Dallinger took microbes from a local pond on the property of the parsonage where he lived, and he started growing them in a homemade incubator of his own design. His choice really was not a coincidence. Darwin spoke of the origin of how the principles of evolution by natural selection would work with microbes, just like with anything else. So Dallinger started these cultures at 60 degrees Fahrenheit, and over the years slowly increased the temperature until he had them growing at 158 degrees. Evolution in action. So Dallinger's work was largely ignored. He published a couple of times, those papers were ignored. I've read them, but they kind of gathered dust for a century. But they demonstrated that evolution can happen very quickly, and also that microbes can be useful for studying evolution experimentally. Now that principle too was lost until the late 60s, when people realized, oh yeah, maybe we can start doing evolution experiments with microbes. And those early efforts then grew in the 1980s into what we refer to as experimental microbial evolution, which is now one of the major ways in which we study evolution. And it makes sense because microbes really are perfect for studying evolution. They grow quickly, so they evolve quickly. They can attain very large population sizes in small volumes. That makes a lot of variation and grist for the mill of natural selection. We can manipulate them easily. We have a lot of tools with which to do that manipulation and then to study how evolution occurs. We also, importantly, can freeze them indefinitely without killing them. E. coli, you freeze it for however long, then you get it warm and happy in a growth medium, it starts growing again. It's fine. You can't do this with zebras or humans. You do it, you get a dead zebra and manslaughter charge. Not good. So microbes, you can do things you can't do with other organisms. And this means that we can maintain a time series of living samples in what we refer to as frozen fossil records of evolving populations. Among other things, this means that we can directly compare ancestors and their evolved descendants in fitness assays. So we can compete them head-to-head in co-culture and actually measure how fitness is changing over time. And in this context, fitness is the ratio of growth rates of one to the other. When I refer to fitness later on, that's what I mean. Now that fossil record also means that we have restore points for when we have to restart the experiment without losing decades of work. So we were able to do that, for instance, when COVID shut us down. We froze the experiment. When the lockdown ended, we were able to start it again. This also means that we don't have to worry so much about making mistakes, because you can't knock something over and end up ruining decades of work. So we can restart. And this also means that we effectively can access, replay, and restart history whenever we like. This frozen fossil record effectively is a time machine. So, as Tim mentioned, I've worked with Rich Linsky since 2004, which I don't know how that happened, but it did. Got my PhD there, stayed as a postdoc, became a research professor. Eventually they gave up with getting rid of me and made me faculty, so they're stuck with me now. And it's been great because, I mean, Rich is the pioneer, the seminal figure in this burgeoning subfield of evolutionary biology. And of course, as Tim mentioned, the centerpiece of the lab's work has been the E. coli long-term evolution experiment, or LTEE, which Rich started back on February 24th of 1988, when he took a single clone of E. coli B strain REL606 and used it to found 12 initially identical populations. And these have since, with very few exceptions, been evolved under a regime in which every day someone comes into the lab and transfers 1% of each to a flask with a fresh volume of a growth medium called DM25 that contains a tiny amount of glucose for food. Under these conditions, the bacteria grow 100-fold every day. That corresponds to 6.67 generations a day. You can think about what that means in human terms. At that pace, over the 38 years of the experiment so far, we've been able to observe over 82,000 generations of evolution in each of the 12 populations. And because we freeze samples every 75 days or 500 generations, we have a complete viable frozen fossil record of the evolution of all 12 going back to the very beginning, giving us an incredible resource for asking a litany of questions, some of which haven't even occurred to us yet. Now, it's important to note this system's highly simplified. There are no invasions, nothing goes into these flasks. It's like they're in their own little space stations isolated from the universe. There's no environmental change, at least none that we allow, though they change the environment themselves. And there's no sex. So there's no genetic exchange. At least the ancestors started off that way. We haven't seen evidence of it yet, but it's something that could happen. It'd be really cool if it would, not just for the bugs, but also for the things that we could ask after that. So evolution in the system occurs by the three core-most processes of evolution. Natural selection, which is relentlessly driving their adaptation. Mutation, which is randomly generating variation. And then a random process called genetic drift, which is just the random fluctuation of variation in the population. Now, because these are the most fundamental forces in evolution, they're going to be true of evolution anywhere in the universe. So this little experiment allows us to understand how evolution is working anywhere in the universe it's occurring. The experiment's also of indefinite duration. And in 2022, Rich handed the experiment off to Jeff Barrick, who was a postdoc in the lab who had gotten a faculty position at UT Austin. Barrick watched over it, and then last year we hired him back to MSU. His wife is from Frankenmuth, so it's fairly easy to persuade him to come up here. Plus, I mean, Michigan's way better than Texas anyway. I think he agrees with that too. So in due course, after some decades watching it himself, he will pass it on to someone else who will hold it and tend it, and then pass it on to someone else, and someone else, and someone else, as far in the future as we possibly can foresee. Now that long-term nature is very important. It means that there is scope for new phenomena to occur, for new things that we never expected to happen. And that expands the scope of the experiment over time. It effectively grows as it goes. It is a model system that itself yields new model systems. And it has a burgeoning community. There are now dozens of researchers who study this experiment and its fossil record and the things that have happened within it. They're scattered across 4 continents, and this community is growing all the time. And we've put some time recently into creating a lab website for the experiment that's acting as a clearinghouse for information about the experiment, protocols, videos, news, publications, and so on. So if you want to know more, go to the lte.org and you'll be able to read about anything you like. There are hundreds of publications, lots to see. So one of the experiment's original goals that Rich had in mind when he started it was to look at the repeatability of evolution. So this is closely related to other questions that regard the role of history in evolution and the role of something called historical contingency. So historical contingency is this tendency of historical systems to be really sensitive to minor variations, to the plays of chance. And it's something that occurs because of an interaction at the core of historical systems between causality, chance, and necessity that make it possible for different histories to occur and for those histories to lead to different outcomes. It, in short, is the property that makes history matter and that makes it so that human history is impossible to predict but explicable in retrospect. And as a friend of mine and great philosopher who studies this area, John Beattie, has put it, history matters when a particular future depends on a particular past that was not bound to happen, but did. So we're all familiar with contingency. It's at the core of counterfactual what-if scenarios that we alternately entertain and torture ourselves with as the basis alternative history science fiction. All these what-if games we like to play. What if the Confederacy won the Civil War? What if Hitler died in World War I? Or more close to home, what if I turned left instead of right that day? What would have happened? So the late great Stephen Jay Gould, who was a fantastic science writer, science historian, evolutionary biologist, and paleontologist, very much a student of human history as well as the history of life, was fond of pointing out that evolution itself has a similar interaction between causality, chance, and necessity as human history. And so he asserted, evolution itself is just as contingent, just as unpredictable, and just as unrepeatable as human history. He wrote about this at length in a 1989 book called Wonderful Life, which did not inspire the experiment, though people often think it's the case. It was kind of a nice coincidence when it came out the next year. And in this book, he talks a lot about these ideas of contingency, and he proposes a thought experiment called replaying life's tape, which he suggests that if you were to go back to various points in the distant past and start evolution over again, then any change in the ensuing history would send evolution down some other path, leading to some other living world than the one that we know and are part of. One of the fond ones, what if that asteroid had missed the Earth and the dinosaurs had not been wiped out? Almost certainly we would not be here. Now, as he pointed out, unfortunately this is an impossible experiment to carry out, but we can do it in the lab. Effectively, the long-term experiment, which is what is called a parallel replay experiment, is an instantiation of replaying life's tape. And it, in fact, is playing the same tape of life 12 times simultaneously from the same point under the same conditions. So we can ask the question, do the minor quirks of history lead the populations to evolve in wildly divergent ways, or do they mostly evolve in parallel, following each other? So in general, we have seen a very complex mix of parallel evolution and divergence. So all the populations have evolved to be much better at growing in their medium. They've gotten to be much more fit than their ancestor. We also see similar broad changes at the level of their phenotypes, their observable traits. All the populations grow much, much faster on glucose than the ancestor did. And they have all evolved cells that are significantly larger and differently shaped from the ancestor. And many of the populations have acquired mutations in the same genes. Now, despite that, they also have their own unique sets of mutations. They're no longer genetically identical. And about half the populations have evolved high mutation rates because of defects in their DNA repair, though all of them have also acquired other mutations that reduce that mutation rate. In 2016, we had this really nifty collaboration with Michael Desai and Ben Good at Harvard University, where they had a ton of money and a lot of sequencing capacity. So they asked us to send them copies of all of our frozen samples, and they sequenced all the DNA in those samples. So all the DNA in the populations, what are called metagenomes. And then they charted out the changing frequencies of the mutations that the sequencing identified. So this is one of the charts that shows what that looks like. Each of these colored lines is a separate mutation that is rising and sometimes falling in frequency. And what was nifty was that some of the populations showed these really interesting patterns where there were cohorts of mutations that were linked to each other. They would rise and fall together, and they would rise and fall in contrapoise with other cohorts of mutations. This is really interesting because this indicates that this population that had started out just completely homogeneous had evolved separate lineages that were coexisting, separate populations. And that is interesting. More so, most of the population show this. 10 of the 12 do. It indicates that ecology has evolved. So diversity indicates ecology, and that's allowing this coexistence. And this is amazing because the tiny amount of glucose that's in the medium, Rich did that on purpose because he wanted to prevent ecology from evolving because it would make in 1988 measuring fitness much harder. It did it anyway, which illustrates Ian Malcolm was right in Jurassic Park. Life does find a way. And also, nature does not like simplicity. She likes complexity, and she will make it from the barest of materials. Now, we think much of this ecology is based on the evolution of very rapid growth on glucose early on that also carries the price of being very sloppy. So like they suck down glucose and then they poop out a lot of acetate or vinegar. And the thing is, once they run out of glucose, they start eating the acetate. But this creates an opportunity. So, acetate specialists in due course evolved that went after the acetate first. So, the acetate users can coexist with the glucose specialists. So this ecology is created by the bacteria changing the environment themselves. Come back to this, because this ends up affecting evolution going forward. So really, whether we're seeing parallel or divergent evolution, it really depends on the level we're looking at. Where we're looking, the genetic level, phenotypic level, metabolic level, whatever. But the populations broadly, we can say, are evolving similarly in many ways. Though beneath those similarities, there are differences. Each population is doing something slightly different, even though they're following paths that are generally going in the same direction. And you can ask, okay, there's subtle differences. Does that really mean anything? Do these quirks of history that lead to these differences add up? Well, one population says that they really can have an effect. To understand what's going on with this, I have to remind you, okay, the DM25 medium has a tiny amount of glucose, 25 milligrams per liter, so tiny, tiny amount. That's their primary food source. But the medium also contains 500 milligrams per liter of citrate, so about 25, a lot more. I'm bad at math, that's why I'm a biologist. It constitutes a potential second food source, this lemony extra thing that we add to help the bacteria take up iron. But the thing is, most E. coli, though they can ferment citrate when no oxygen is present, they're unable to grow on citrate when oxygen is present. This is to say they have a CYP- phenotype. And this ultimately comes down to being unable to make a protein called a transporter for citrate to bring the citrate in the cell. Basically, when oxygen is present, E. coli has no way to eat citrate. It's not food to them. And this trait is in fact very stable. In the entire 20th century, only one spontaneous mutant that could aerobically use citrate, so CYP+, was reported. The entire 20th century, during which E. coli was easily the most intensively studied organism in history. But in early 2003, just after 33,000 generations, one of the 12 populations, which we call Aero-3, suddenly got much bigger, much cloudier. And it turned out that population was full of E. coli that were growing on the citrate. Now, I went into the frozen fossil record when I came to the lab the next year to try to figure out when it first arose, and it turned out that weak CYP+ guys were there in the population as early as 31,000 generations. But they were so weak they weren't really effectively growing on it. But natural selection did its thing, and over the next 2,000 generations, they acquired mutations that made them better at using the citrate. Strong citrate users arose, and they took over the population and clued us into their existence. So CYP+ is what can be called a novel trait. So novel traits are qualitatively new traits that when they arise confer new ecologically relevant traits on the organism. They've been a major, major factor in evolution because they allow organisms to start accessing and using resources they couldn't before. They allow them to invade new niches, and in so doing, they increase the amount of ecological diversity, they increase complexity, and they start driving speciation. And they can in fact be quite revolutionary because they change the conditions under which later evolution takes place. So if we think about novel traits, the canonical ones that we tend to think of, sight, flight, photosynthesis, multicellularity, they completely change the course of history on Earth. So CYP+ is like a minor version of that. And certainly, we see the massive consequences a novel trait can have by looking at CYP+. It caused the population to get 10 times bigger. It ended up eliminating most of the diversity in the population, leaving only the CYP+ lineage and a CYP- lineage that didn't use citrate that was able to persist for about 10,000 more generations. So it completely changed the ecosystem, and for whatever reason, it caused increased rates of cell death. We have no idea why, and natural selection doesn't seem to be knocking that back. It's a big mystery. We're still figuring that out. Maybe in 5 years, I'll be able to come back and tell you what's going on there. We'll see. There's a great PhD student in Illinois. He's going to be looking specifically at that question with my colleague Nkrumah Grant. But we're also seeing over time evidence of incipient speciation in the CYP+ lineage. So we're seeing it get much better at growing on citrate, while at the same time it's losing the ability to compete for glucose, so the ancestral resource. And they're doing so in lockstep, which indicates the same mutations that are making them better at citrate and worse at glucose. These end up being barriers to gene exchange between CYP- and CYP+, exactly what you would see in speciation. So CYP+ really is this great opportunity to study a novel trait that has arisen in a model system that is well characterized, for which we have a lot of tools, so we can study its origin, its evolution, its consequences, and so on in fine detail in ways that we can't with others. So the big question about CYP+ that I took on when I first came in was, why did CYP+ take so long to evolve? It took almost 2 decades to do. Why has it only evolved once in the long-term experiment? And it's still only once. 50,000 generations later, no other population has done this. So what's going on? So I posed a hypothesis of historical contingency, that what happened was this population evolved CYP+ because over the course of its particular history, it saw the accumulation of mutations that increased the potential to evolve CYP+. They potentiated its evolution. And I was able to test this Gouldian hypothesis in a Gouldian way. So I went into the fossil record and I took out 271 clones, so individual cells from 14 different time points, and I replayed the tape of CYP+ evolution and looked for the pattern with which CYP+ re-evolved. So basically the idea was it should be easier to re-evolve CYP+ after those potentiating mutations were in place. And so we should see more re-evolution of the trait after they were in place in later time points. So if you want to think about this in another way, say the MSU Department of Experimental History were to hypothesize that the Allied victory in World War II was potentiated by Germany making the idiotic decision to invade the Soviet Union. So they test this by getting in their TARDISes, going back and replaying World War II over and over and over again from various points, and seeing how often the Allies won when they restarted from that point. And they would expect, if they're correct, then the Allies would win more often after that decision was made to invade. Same thing without the death or time machines, or at least not a TARDIS. Oh well, someday. I'm accepting grant money. So over the course of that experiment, I tested a total of 40.4 trillion cells. It was not a trivial undertaking. If you want to get a sense of that, this is what the aftermath looked like. I am obsessive compulsive. I did count. That's 13,704 petri dishes. It was not the only tower because there were follow-up experiments. When I die, these towers are what I'm going to be known for because they're in textbooks. But all that work paid off. I saw CYP+ re-evolve 13 different times, and each time from replays that were started with clones that I took out of the fossil record from generation 20,000 or later, and that pattern strongly supported the contingency hypothesis and argued that something happened around and then after 20,000 generations that made it much easier to evolve CYP+. So what happened? Okay, that's an incredibly complicated story that I tried to unravel and wasn't able to, and then my colleagues Eric and Dasha working with Jeff, their PhDs were working that out, and we still don't think we have the full story, but to simplify what we think happened was, early on an acetate ecology evolved like I talked about earlier, and the acetate specialists took a particular and unique evolutionary path that occurred in none of the other populations. And over the course of this, they acquired unique mutations that didn't occur in other populations that had the effect of not only improving their ability to grow on acetate, but also coincidentally their ability to grow on citrate before CYP+ evolved. So with those mutations, it had an interesting effect. With those mutations in place, CYP+ became slightly beneficial. Without those mutations in place, if we make the cells CYP+, they're very bad. They're very detrimental. They die quickly. So, effectively, this particular way of adapting to acetate pre-adapted their metabolism for growth on citrate before access to citrate evolved. So, what was the mutation that caused that switch from CYP- to CYP+ and gave access to citrate? So to answer this, we did a pretty big program of genome sequencing of clones from this population at different time points. And basically we asked what happened right there where CIT+ evolved. And going through the genome sequences, we found that all of the CIT+ clones had a duplication mutation. There was a 1733 base pair segment that was duplicated in all of them. These are in a tandem head-to-tail orientation, so copy, copy. And this duplicated segment overlapped part of the citrate operon. It contains the genes that are coordinately regulated that control citrate fermentation, and it's centered on a gene called CYT. CYT encodes a citrate transporter. And the way the duplication worked, it put a copy of CYT under the control of a switch called a promoter that tells the cell when to turn the gene on, that normally controls a gene called RNK that's turned on when oxygen is present. So in its new location, this switch told the cell, make CYT when oxygen is present, and that gave access to the citrate. So this is a rewiring of the genome by this duplication, creating what we call an RNK CYT module. So this is very much an example of what the great molecular biologist Francois Jacob called evolutionary tinkering or bricolage. Because in evolution is kind of lazy. It doesn't like to create from anew. It basically tries recombination of stuff to see, and if something has a function that's useful, it continues. As Jacob put it, evolutionary novelties come from previously unseen association of old material. To create is to recombine. But there were consequences to using this transporter. CYT is what's called an antiporter. This means it powers the import of citrate into the cell by pumping something else out. From inside the cell where it's at high concentration to outside where it's low concentration. This is a principle you all learned in biology class and promptly forgot. I did that too. They beat it into you eventually. So in the case of CYT, what gets pumped out are these things called succinate fumarate malate, which all you need to know is these are metabolic intermediates of citrate. They're partially digested citrate. So basically, it's sucking in citrate and spitting a lot of its food out. So it's losing a lot of the value of CYT+. It's kind of like a baby learning to eat spaghetti. Probably 90% of the spaghetti ends up on the baby and on the high chair for every little bit that goes in. Same principle. In order to get that food that it spills out, it has to use another transporter called DCTA. And it is what's called a symporter. So it can bring those lost food resources, succinate fumarate malate, into the cell by allowing protons to flow into the cell with them. The problem is, this isn't expressed in the ancestor because the regulation of it's broken. But in due course, by 34,000 generations, the CYT+ lineage acquired another mutation that caused this DCTA transporter to be produced. So it was able to recover its lost food and really finally get the full benefit of growing on citrate. This also made growth on citrate more efficient because it brings that succinate fumarate malate back in, and then it can pump it out for more citrate. So it just makes it much easier. So this is really odd when you think about it from an engineering standpoint, because in order to get good at growing on citrate, you need both CYT and DCTA, which is really Rube Goldberg. But Rube Goldberg is a patron saint of evolution because biology is just full of all of these really kind of dumb engineering compromises, stupid, you don't want to call them decisions because there's no mind behind it, and silly workarounds. We find them all over the place. We have aching backs because of this, for instance, but it's all over the place. So it couldn't just be these transporters, though, because there are genes that regulate metabolism that is being used to break down the citrates. There were other genes that were involved that had to be there in order for the expression of CYT and then DCTA to allow growth on citrate. What were they? How many were they? This is what I call the deep genetic basis. So this is a project that was undertaken by Tanush Jagdish, who was a student at Kalamazoo College, who saw Rich speak and begged to come into the lab. He said, sure, this is a project that no one has actually really succeeded at. Will you do it? And he did. He took what's called the Keogh collection. So this is a painstakingly made collection of E. coli strains where they have gone into 4,000 of them and knocked out a single gene in every single one of them. So you can see the effect of knocking out each individual gene. And what Tanush did was he took a plasmid, a small circle of DNA that had an RNK-cysteine module on it, that if you put that plasmid into the ancestor, it makes it CEP+. So he put that plasmid into all 4,000 of the Keogh strains, all 4,000. And he looked for those that with the plasmid stayed CEP-. Those are the ones where the gene, a gene that was necessary for citrate use was knocked out. He expected maybe finding 10, 12, that was my guess. As it turned out, he found over 100 genes that were necessary to grow on citrate. So that deep genetic basis of CEP+ was really very extensive. And what this reflects is that effectively the genome of an organism is like a junk drawer. It's filled by past evolution with all these odds and ends, and the long-term evolution experiment's ancestor, its history left it with this rich junk drawer of elements that, they were beneficial at various points in the past, even if they weren't under the conditions of the experiment, but that information, once it was there, it could be recombined, it could be tinkered with, and yield new traits, including CEP+. So CEP+ evolution was contingent not only on the history of this population in the course of the experiment, but also what happened before the experiment. And CT was the gene that happened to be in the junk drawer. It was the gene that had been used in the past for citrate fermentation. So the easiest way to evolve CEP+ was to use CT by activating its expression, even if it's not great for that job and requires this Rube Goldberg mechanism. This reflects how evolution, like Jacob suggested, builds from the legacy of past evolution and what's already there. Now, that didn't have to be the case. CT is not the only citrate transporter. There are relatives of E. coli that have other citrate transporters that work in completely different ways. So one is called CYTA. It is a symporter, so it brings citrate in by letting protons in. There's another CYTS that brings citrate in by letting sodium in. So they don't need another transporter like DCTA to make full use of the value of citrate. So we can ask the question, what if the long-term evolution experiment ancestor had those genes? What if its history put that there instead of CT? What if CEP+ had evolved based on them? How would the evolution and the consequences of the trait been different? This is a little like asking, how would birds have evolved differently if they had wings like those of bats or pterosaurs instead of birds? As you look at the different kinds of wings that evolved, they all perform the same function of helping with flight, but they differ in their details, exactly how they work. And there are things bats can do that birds can't, and vice versa. So would these have had different consequences on bird evolution with these different forms of wings? So we have issues, things like this in human technology and history. So those of us who lived through the 80s remember Betamax versus VHS. One of my favorites is with automobiles. So around 1900, there were automobiles that were steam-powered, they were electric, there was gas-electric hybrid, and in fact, the earliest car was electric. And then there were internal combustion engines, which were regarded as novel death traps because they would just blow up randomly. Everyone expected, we know how steam works, we know electricity, those are what's going to win. But they didn't. Internal combustion won out. So we can ask the question, what if those other vehicles with other transportation technologies had become established as the standard? How would environmental history have been different? How would geopolitics have been different? How would economics be different? Well, these are counterfactuals that are only thought experiments. But even though these are just thought experiments, we can make counterfactuals effectively real in evolution using E. coli because we can genetically engineer them. And I'm currently working on starting a project that's going to do just that, where I'm going to recreate this population with alternative versions of CEP+ that use these other transporters, and then see how the consequences and evolution of CEP+ change these other bases. So ask me back in 5-10 years and I'll tell you what happens with that. So going back to the whole genomic junk drawer idea. So natural selection is often seen as an optimizing algorithm. It's making everything better, daily and hourly scrutinizing, adding up all that is good, subtracting all that's bad. We see it as increasing efficiency, getting rid of what's unnecessary. But the thing is, the genetic information that natural selection works on comes from ancestors, from past evolution. That information at some point was beneficial. It was selected for. It helped adapt those past organisms to their environment, but that past adaptive value may not correlate to current adaptive value. Effectively, a lot of the genomic information in a genome, including ours, is unuseful. It's junk. And yes, over time, if a gene or genetic information is not of fitness benefit, over time it gets lost. Because evolution is not forward thinking, it's not forward looking, it's not planning ahead, and it doesn't retain things just because they might be useful. If there's nothing keeping selection to keep these, they're lost over time. But there's evolutionary inertia. Those inherited traits, that inherited information, it's maintained for a long time, because it turns out this degradation, it does not occur very quickly. We still have the remnants of genes necessary to make vitamin C, even though our ancestors lost that millions of years ago. Whales still have the genes for olfactory receptors, even though they lost those tens of millions of years ago. So genetic information will linger in the genome for a long time, and that means that it remains available for tinkering and recombination, and that can lead to the evolution of new traits, including novelties that can bring radical revolutionary change. So this is part of why evolutionary history matters. Evolution never starts with a blank canvas. There's always, because of inertia, this junk that's in the genomes, this legacy of past evolution. So the past lingers, and it alters what can evolve in the future, because the future is built on top of the past, and the past doesn't get overridden quickly. And this resistance to change maintains the capacity to change. It's called evolvability. Evolution would be slower if unneeded genetic information were lost immediately after it's no longer beneficial, if there's constant ready streamlining. So this means the genomic junk drawer doesn't get emptied very quickly, just like one of our junk drawers. And we see the effect of this in the long-term experiment with CIT+. CIT, that gene's normally not expressed. It was actually of no benefit at all in the long-term experiment. It could have been lost at any time with no harm to the fitness of the organism. DCTA2, that gene is inactive in the ancestor because its regulation is broken. And so it too could have been lost at any time with no harm to fitness. But they weren't. That evolutionary inertia that makes it difficult to just break down and get rid of this stuff, it meant that those materials needed to evolve CIT+ stayed in place. So to go over the bare findings that I've talked about, the evolution of the CIT+ trait in the long-term experiment shows how chance history and quirks can make it possible to evolve revolutionary new traits. The trait arose because of a duplication mutation that created a new combination of genetic information that is a perfect example of evolutionary tinkering, that this was set up by the evolution of a particular ecology, and that how the CIT transporter works meant that in order to get good and efficient at using citrate, there had to be the expression of another transporter entirely, and there were a lot of genes other than these two that were necessary for the trait to manifest. And the history of the ancestor of the experiment created a collection of genes that made it possible to evolve this trait. So the takeaways are, Darwin's legacy is still going strong, still growing. Microbial experiments, like the long-term experiment, are awesome, because they let us study evolution in action. The long-term experiment itself is just getting more interesting over time and allowing us to study more and more aspects of evolution. History's quirks really can matter a great deal in evolution. As history changes, what can evolve? Selection acts on the variation that arises from modification of the genetic information that's there because of prior history. History stocks this junk drawer of the genome with genes and materials that can be recombined and tinkered with to create new traits. And genetic information that's not of immediate value can be the stuff that game-changing novel traits can be built from. So evolutionary inertia ensures that genetic information, this junk, is not rapidly lost immediately after it's no longer beneficial. It keeps the genomic junk drawer from being cleaned out quickly, and that maintains evolvability. So evolutionary inertia can lead to revolutionary change. And I think that there is a lesson for us in this age of streamlining that comes from evolution about the value of not throwing away things the moment that we think that they're no longer useful. And just-in-time production is maybe not a good idea. We would have thought we would have learned that from COVID. And finally, I thank my lucky stars that I get to do what I do, that I get to study evolution, because really, Darwin was right, there is grandeur in this view of life. So right before I end, I want to just say thank you to all the collaborators I've been fortunate enough to work with over the years, and the undergraduate mentees. It's been the greatest honor of my time in science to work with these fine people who did a lot of the work that I took credit for today, FYI. And of course, NSF, the Beacon Center, DARPA, Kenyon College, and the Templeton Foundation for funding it. So, thank you.
Thanks, Dr. Blount. That was a quite interesting and informative lecture. We're going to do the Q&A time now, and we only have one mic up here, so you can raise your hand if you have a question, and Dr. Blount, maybe you can just point people out, take questions from, and then repeat the question so that we can get it in the recording.
Okay, sounds good. Okay. Yes.
Am I correct in thinking that what you have been observing is drift and not a controlled change in the environment?
So the question is, have I been observing drift and not a controlled change in the environment?
A manipulated change?
Oh, definitely there's no manipulated change. We just provide the stable environment, the temperature, the shaking of the flasks, and the medium. So we're not exerting any conscious selection on them. And as I said, the bacteria are changing the environment themselves, so if anything, they're generating their own selection. Yes, it's not an artificial selection experiment.
You had mentioned speciation is a little bit different for, I guess, how does speciation work if there's no sexual reproduction?
The question is, how does speciation work if there's no sexual no sexual reproduction? That is a gigantic question. No one agrees on speciation with sexual organisms, much less asexual ones, and everyone is certain that their particular model is correct, and you will have people yelling at each other red in the face at conferences over this. It's very complicated, and it says something about the complication that all of us have Neanderthal genes. Neanderthals were a different species, so clearly that whole thing that species can't reproduce with each other, that's not as true as we once thought. Dogs, wolves, coyotes can all interbreed, for instance. There are open questions about whether or not we are actually completely reproductively isolated from chimps, despite several million years of separation. Now, with bacteria, there are well over 100 different concepts. No one agrees on this. The one that I like the best is one that's articulated by Frederick Cohen, who's a professor, a really nice guy at Wesleyan College. And his idea is speciation occurs in bacteria when a mutation occurs that allows a lineage to shift into another niche so it can coexist with its parent lineage and undergo what's called periodic selection. So it is accruing beneficial mutations that lets it get better in its new niche, but does not encroach on the parental niche. And so they're able to coexist long term. In his opinion, that's speciation. There are a lot of people who disagree, who say, well, that's just too easy. We're not going to be able to name all of those. And his point is, why should nature care what's easy for humans? And I tend to agree with Fred's idea, and kind of when we say that we're seeing incipient speciation in a long-term experiment, we're using his ideas. Now, I've also been doing work where I've been taking mutations from CIP+, that improve fitness on citrate but reduce fitness on glucose, hypothetically, and putting them into CYP-. So if they are like this, if they have this differential effect, then we put them into CYP- and they're bad, but in CYP+ they're good. That effectively, if there was sex between these, would be the rise of incipient barriers to reproduction. And in nature, bacteria engage in effective sex all the time, though even there they don't respect species barriers because they archaean bacteria that have been separated for billions of years. They exchange genes all the time. We find bacteria with human genes, humans have bacterial genes. None of that barrier stuff is, nature doesn't care, basically.
What originally got you into biology?
What originally got me into biology? So, I grew up in the country in North Georgia, and I was out in nature a lot, and was just enamored of it. And my mom, when I was little, got a master's in biology education, and I remember bath time when I was like 3, and she would be studying by singing songs of her study materials, and I kind of think that maybe that got inserted into my head. Now, I was interested in biology, and in middle school I read a book on evolution and got really interested in evolution, but I was bound and determined I was going to be a physicist because I loved physics, and I was going to major that in college, and I got to college and I discovered, no, I could not just will myself to make it through calculus. And I realized, okay, I don't math so good, I'm going to go to biology like Ms. Pugh, my AP biology teacher, told me I should, and she was right. So listen to your teachers. And your mom when she's singing you songs about biology. Yeah.
I have a follow-up on the speciation question. Have you all discovered that any of the generations are a different species than the first ones?
Have we found a point after which there are different species than the ancestor?
Mm-hmm.
That is a very dicey question. Under Fred's model, we could argue that they are different species, and Rich and I have bandied about the idea of calling the CYP+ guys Escherichia gouldii, but we're not bold enough to actually go out and state that claim because we would face just fiery breath from people who disagree with Fred. So I think a strong argument could be made that we've seen separate species evolve in most of the populations, because those instances of ecology and stable coexistence of multiple lineages, that's indicative of speciation. And I think that we've seen speciation in at least 10 of the 12 populations. But making that argument in a way that other people would believe, that's a difficult thing, but I would say nature believes that they're species. Yeah. And she's the one who matters.
Another question.
Okay.
This is related to freezing the bacteria. My understanding of cryogenics is it doesn't work because we can't freeze human cells because then the cell wall gets ruptured from the frozen liquid. But apparently bacteria, that does not happen. Why is that different?
Okay, so why can we freeze bacteria but we can't freeze humans? A lot of it comes down to we're much more fragile. We don't have cell walls the way bacteria do. And you know, when we freeze our samples, we add a cryoprotectant called glycerol that prevents the formation of large ice crystals, which usually do the disruption. They're tiny little crystals. But we found that even if we freeze them without cryoprotectant, and there are frozen bacteria all over the environment that freeze without the benefit of cryoprotectant, 99% of them survive once they get warm again. They're just really hardy, and there are a lot of them. If you have billions and billions and billions of cells, if you wipe out a billion, it's no big deal. You wipe out a couple billion human cells, you start running into problems, especially depending on where they are. So they're just really hardy, and there are a lot of them. And they have, in their histories, probably gone through freeze-thaw many, many times. So you think about E. coli, how E. coli gets around. It's in all of our guts. It gets pooped out in the environment. It's in the guts of all mammals and a lot of reptiles and birds. It gets pooped out in Michigan. Michigan's quite cold. They freeze, they thaw, they're fine, they move on. And as I was telling Tim to begin with, we've isolated, not we, but scientists have isolated bacteria and gotten them growing again after millions of years of being frozen in permafrost. Just, they're hardy, they survive.
All right. Thank you again, Dr. Hall. And like we said at the beginning, if you are interested in gathering, we're going to go off to the Palace of India. Otherwise, we will see you at another event some other time. Thank you for coming out.